is a three-parter, and they each have names: presumptive, confirmed, and completed tests. We have to know the names, I'm sorry to tell you. So the presumptive, the confirmed, and the completed tests. So there's 3 tests for the coliform bacteria, and coliform, that also has a definition which we do need to know. It means having E. coli-like characteristics, and that means that you're just like E. coli. If you're a coliform, you are gram-negative, you are bacilli, you don't make spores, so you're non-spore forming, and you ferment lactose with acid and gas production. Okay, so E. coli is prevalent in the lower intestine of humans. It's about 1% of the bacteria found in your feces. And let me tell you, there's a ton of bacteria found in your feces. E. coli is only 1% of it. So what's the other 99%? Well, that depends. What do you eat? What do I eat? Where do you live? Where do I live? Do you have pets? What are my pets? Okay, what meds are you on? What meds am I on? How old are you? How old am I? Okay. All of these things influence the composition of what your normal microbiota might be. So I can't agree with you on the other 99% of bacteria found in feces, but we all have E. coli. And so because of that, E. coli becomes the test organism for feces. E. coli is typically not harmful. It's actually one of your friends in your lower intestine, okay? Normal microbiota. But because it is found in human feces, commonly found, everybody's got it, I can test for the presence of E. coli, and that would tell me that feces is present. So we test water samples for E. coli in the coliform analysis to see if that water sample has feces in it. I am not concerned necessarily with the E. coli. In most cases, E. coli is no problem whatsoever. Of course, there is the E. coli 0157. Every once in a while, you hear about it. I think the last time, I think it was in spinach. Sometimes it's romaine lettuce. It was melons not that long ago, probably 6-8 years ago at this point. There is a bad form of E. coli, but typically I'm testing for E. coli not because it's the harmful one. What I'm worried about is if there's E. coli in the water, that means there's feces in the water. And now I'm worried, like, what else is in the water? Because we're going to find out in our pathology, pathogenic bacteria lectures that fecal oral contaminants are plentiful. There's a lot of them. So in this lab exam, I will ask you to name 4 fecal oral diseases. I will name 2 for you right now, and those 2 cannot be used. One of them is cholera. You know about this already from the video that we watched independently. We watched them at home. That cholera, and remember they cleaned up the water source, and by doing that they got rid of cholera. Cholera is fecal oral contamination. Another one is polio. Polio, teeny tiny virus, naked RNA, right? Really, really small virus. That is fecal oral. It's super hard to find virus. You know that. It's so small. And then I can't isolate it and grow it that easily. I have to put it in a tissue or in eggs. This is a big problem. I can't find that very easily. But what I can find easily is E. coli. E. coli would tell me there's feces in this water, and then that water becomes undrinkable or unavailable for recreation. No swimming in this beach, no swimming in this pond, etc., because of what else might be in there. Okay, so it is a 3-part test. The first one is known as the presumptive test. And the presumptive test, see what that means? presumptive. I already assume, I presume it's in here. Guys, so let's just like assume that this water sample is contaminated. So I would have asked you to bring in a water sample, and the best water samples to bring in are like well water. In fact, the coliform test that we do is the exact same one that the county does on your well, on your property if you have it. And so typically people would bring in well water from their house, and from the tap in the house, and well water from the well itself. And that's kind of a good test, because then you could see if the filtration is working. You can also bring in pool water, like a public pool. You can also bring water in from a beach or a pond or a river or a creek or whatever. Those were the samples that I would ask you to bring in. And okay, so we would test this water sample, and we would do it in 3 different tubes. Okay, the tubes would have lactose broth. That would be the only thing in here. It'd be lactose and water. Okay, lactose is a sugar, and it's a food source. Okay, lactose broth, it is the sole carbon source. That's the only thing to eat, in other words. Okay, all organisms are carbon-based. All 4 carbon compounds have carbon in them, don't they? And so they need carbon and carbon sources. And in this case, this is all there is to eat. So if that's the stringency, then this test is already selective, isn't it? If you cannot metabolize lactose, you cannot live in here. So it's selective, and this is why lactose is the sole carbon source. There's nothing else. There's no protein in here. There's no lipid. There's no nucleic acid. This is it. Can you survive on lactose alone? That's the question right here. And so we would take your water sample, and we would put 10 milliliters into the first, then 1 and 0.1. And this is kind of a way to do a little bit of a qualitative and quantitative analysis of your water sample. Remember, to be a coliform, not only would you ferment this lactose, but you would also produce acids and gas. So acids, I am not going to be able to see. I don't have phenol red in here or anything. There's no acid test. But I can check for gas with something called a Durham tube. A Durham tube is a small glass tube that has the opening at the bottom, and the closed part goes on top. So you sort of put it in there upside down in all 3 tubes. You would add the 10 milliliters of your sample to the first tube, 1 milliliter to the second tube, and 0.1 to the third tube. And we would just incubate it overnight and see. So what kind of results would you get? Well, so we can see that in these tubes, all of which are lactose broth, okay, so this is the presumptive test. Notice that in this case, this is the way I put it together anyway, that I would have some turbidity. It's not clear in this tube, indicating growth. But more than that, I have gas in the Durham tube. I have it in the 10 milliliter tube, the 1 milliliter tube, but the 0.1, I don't see any. So remember, I'm looking for the presence of E. coli. So it looks like it might be in these 2 tubes that both have greater concentrations of your original sample than the last tube, which has a tiny amount. So if you do have a fecal contaminant going on in your water sample, your well, your pool, whatever, it might not actually be that bad, because if it were a really bad situation, it would be present even in the lowest concentration. So I hope you understand that. So in the presumptive test, the only way to pass is with gas. Gas is the pass. So this last tube, the 0.1, that's a fail. There's no gas there. That's a fail. Okay, so for some people, if we had done this in lab, some people, you come in with your well water, gee, I hope everything is fine. And, all 3 tubes would have nothing. No gas in any of them. And you could leave and say with certainty that there's no E. coli and therefore no feces in that sample. Okay, those people would be happiest of all. This is one of these tests you want to fail. Other people come in and they do this and, oh, I have gas. Oh, I have gas. I think I may have feces in my water. I can't know for sure yet, though. This is a 1 of 3-part test, okay? We would go forward only with tubes that have gas in them. So if you were somebody who's all 3 tubes showed up and you were negative, you'd have to borrow somebody's to go forward, okay? To go forward, we would take a sterile loop full of the sample that has gas, but the least amount of samples. So which one would I choose here? You have to have gas, but the least amount of samples. So yes, it would be the 1 ml sample. In this case, we would go forward with 1 sterile loop. So then we would go on to the confirmed test. So it takes like a while to get through the coliform analysis because it's 3 tests. So the confirmed test would take this positive sample that had gas but the least amount of water sample in it. In our case, it was the 1 ml. You would take a sterile loop and you would put it into the brilliant green lactose bile broth. Boy, what a mouthful. Brilliant green, that's the dyes in there. Lactose, still the sole carbon source, and there's bile in here. Okay, brilliant green lactose bile broth. It is a beautiful color. We are really missing out because, and I should stop saying that, I'm sorry. We're just making the best of what we have, but it's like this emerald green. It's so pretty. Anyway, it also has a Durham tube in here, this upended glass tube with the opening at the bottom and the closure at the top, so sort of upside down. You would take a sterile loop full of this and put it in here. The brilliant green dye and the bile both inhibit gram positives because in this first test, okay, with your lactose, you're looking for a lactose fermenter with gas production. And what I can see with gas is I have fermentation that has resulted in gas production. But I don't know if it's on gram negative. And to be coliform, you must be gram negative. So I'm going to inhibit gram positives with the brilliant green and the bile. Both of these, the dye and the bile, inhibit the formation of peptidoglycan. Peptidoglycan, you remember, is the cell wall, and gram positives have a very thick cell wall, and that is their outermost layer. If they cannot make a good solid cell wall, they will die. So they will not survive in brilliant green lactose bile broth. So we put 1 loop full of this lactose broth culture with the gas. Okay, your water sample that gave you the gas. Put 1 loop full into brilliant green lactose bile broth and incubate 37 degrees overnight. And then it would be coliforms part 3 and 4 would be the following week. We can only go forward with gas. Gas is still the pass. Okay, so if we look at these tubes here, what do we see? Which one has gas? Only tube number 1. So this, you would only have gone forward and done 1 tube. I'm just saying if you looked at 3 people's tubes together, say. Only gram negatives can survive here. So this tube here, I don't see any turbidity or change or whatever. So whatever was in, now remember, the only reason I could have gone forward is if I had gas. So if I had gas and went forward into the brilliant green lactose bile broth, but now I have no gas, then I know that whatever gave me the gas before must have been a gram positive, and now I just got rid of it. With the bile and the brilliant green, I inhibited the ability of that gram positive to make a cell wall, and so now they're dead. They did not survive in here at all. Okay, I couldn't have gone forward if I didn't have gas. All right, what about this guy? Well, this person went forward with gas, and now I see turbidity, but I don't see any gas anymore. This is a fail. So there is definitely growth in here. So that means there's gram-negatives doing just fine in here, but whatever gave me the gas before, that must have been the gram-positive because now it's dead. And now I don't see any gas anymore. Both of these are over. So the person whose well water came up positive in the presumptive test in the lactose broth, very concerned, oh my gosh, there's feces in my water. And I would say, hey, you don't know yet. Here, at the end of this confirmed test, they would go home happy knowing that whatever is giving them gas in the lactose broth was a gram-positive, which means this is not fecal. It's not E. coli. It's not fecal. And so what you would know is you have bacteria in your water, and you probably do. Okay, bacteria are everywhere. So unless you literally sterilized your water, yeah, you got bacteria in there. That's just the way it goes. All right, that brings us to tube 1. Tube 1 has, first off, color change, which lets me know some acid has been produced and the brilliant green has changed color. But a change of color is not the pass. What's my pass? Oh, that's right, gas. Gas is my pass. So here we go. I have got gas here. I have now, what do I know about tube 1? I've got a gram-negative because I have survival. And not only is it gram-negative, it is a lactose fermenter with gas production. Oh, that's awesome. This is my pass. I mean, it's not awesome if it's your well water. Now you're really concerned. You know, for most people, their wells at this point have failed. And usually the only people who are moving forward are people who took water from, like, you know, a fountain or a creek or something like that, but okay, it could have been somebody's well. All right, we go forward. We go forward with this tube. And so what would happen is that this person that has the gas in their tube would be having to loan their tube to a bunch of people because most people are going to fail. All right, so we would go forward with the tube with the gas. And now we would take one sterile loopful of that positive gas, okay, so positive confirmed test, positive brilliant green lactose bile broth, the positive is the gas. Take one sterile loopful and we would just streak it out onto eosine methylene blue agar, EMB. Now, we just did mannitol salt a minute ago, and it might look from these images that they're the same. They're not. This EMB is actually more purpley, and mannitol salt is more orange pink. So I'm sorry if they seem like they're the same. If you saw them side by side, you would not confuse them. So eosine methylene blue has two dyes, eosine and methylene blue. Eosine is red, so you've heard of eosinophils and stuff. So eosine is red, methylene blue is blue. Together they make kind of a purpley red. It's more red than purple, but still. Both of those dyes inhibit gram-positives from growing, so I still have a selective situation going on here, okay? And if there's E. coli in here, I will get a really distinctive result. This is one of my favorite labs. But this one I really like. Eosine methylene blue is taking advantage of the fact that E. coli produces a ton of acids when it ferments compared to other gram-negatives. So what's in eosine methylene blue agar? You don't need to know the specific composition, like the percentages, because that is just too hard to keep that in your head. But there is peptone in here. Peptone, okay, remember that's proteins. There's the agar, that's indigestible. There's now two fermentable sugars, not just lactose, but also sucrose. So if they can ferment sugars, they've got lots of sugars to work with, which will give me lots of acids on the other end. And then I have two different dyes, the eosine, which is red, the methylene blue, which is blue. So the two sugars provide lots of fermentation opportunities, okay? And moving forward, we remember that E. coli, if we looked at this slide, that E. coli would actually produce a lot of acids. So it would produce lactic acid, succinic acid, and acetic acid, which is like vinegar, right? So we've got three acids produced here versus enterobacters, which are very common. They're in soil, which is, you know, soil's everywhere. It could easily be in your well. It's definitely in a creek, right? Okay, so enterobacter makes ethanol just like E. coli does ethanol. That's not acidic. Lactic acid, okay, same lactic acid. Formic acid, okay, formic acid. Butanediol, acetone, these are not acidic. Those are not acidic end products. Carbon dioxide is not acidic, and neither is hydrogen gas. So ethanol E. coli makes way more acids than other gram-negative bacteria. So E. coli is going to produce a ton of acids. EMB agar is designed to find E. coli. And so remember that E. coli is the test organism because it is 1% of the bacteria found in human feces. So that's what we're really concerned about. So we're going to be the ability of E. coli on EMB. And E. coli will give you these distinctive metallic green colonies. It's incredible. You cannot mistake this. These are the best tests when you just look and it's like, yes, there it is. Not like you're rooting to get E. coli, okay, in your sample, but it's not one of these tests where you're like right on the cusp. I don't know. Is it a yes? Is it a no? This is so obvious. You will get a metallic green colony for E. coli and E. coli only. And the reason is all those acids that E. coli makes, lactic acid, succinic acid, acetic acid, react with the eosine and the methylene blue to give you this distinctive color, this metallic green. Enterobacter aerogenes is a gram-negative bacilli which also ferments lactose and does make lactic acid, but it doesn't make as many acids as E. coli. It will grow on this surface of this agar, no problem. That's not going to be a problem for it, but it's going to look sort of like purpley. Sometimes people say sort of a mucusy color. It's not this, okay? It is not the metallic green. So when you get that, then I come around and tell you that you have dirt in your well or dirt in your water sample, and that's typical. Hey, dirt's everywhere. So once again, I'm looking at EMB here, which has two special dyes that react specifically with E. coli. Remember that EMB agar has two sets of sugars, lactose and sucrose. And remember, sucrose is a disaccharide. So is lactose. There's actually, really technically 4 monosaccharides in there, right? And so there's a lot of fermentable ability there. And so E. coli will give you a lot of acids from that, giving you this metallic green. Other organisms don't. And the typical other organism that you might find is Enterobacter aerogenes. Now, Enterobacter aerogenes is one of the 8 organisms that I would have given you in the unknown. And this reaction on EMB is something to take note of because you might get as your unknown EA, Enterobacter aerogenes, which would have passed all the way through the coliform analysis, but here, oops, it's not E. coli, right? It's different, but it is a result that we can work with and we can remember.